Well, thank you so much. What I will try to do today is give you a brief introduction in what we think can happen over the next 10 years if we pay attention to the health of our cells. Every one of our bodies is made up of some 10 to the 13 cells. And what makes cells work are the marvelous machines, molecular machines that are encoded in our proteins. So proteins, usually if you Google protein, you just end up with food websites and nutritional substances. But the reason why we eat proteins is to take them apart and build proteins in our cells that then can carry out these marvelous functions. So proteins function as enzymes. They convert metabolites and food substances. They are the motors that exert mechanical forces to move things about and to move inside the cell and the cells themselves. Structural proteins function as mechanical devices to give cells their rigidity. Then we have receptors and signaling proteins that receive information and allow cells to communicate with one another to have an organized organism. Gene regulatory proteins tell the genome which gene to express when and where in an organism in a cell-specific fashion, and so on and so on. Every one of our cells makes about 1 million to 1.5 million proteins every hour. So it's a huge process going on in the cell. We've learned a lot about protein synthesis. The ribosome translates the copy of the genome made in the messenger RNA, and it translates it into a linear sequence of amino acids. And once these amino acids are polymerized to one another in their specific sequence, they are an unfolded chain. And this cartoon here shows us the length of this chain that then has to be folded up into a three-dimensional shape, so it can carry out this function. And this is the degree of compaction drawn approximately to a scale here. The reason for this compaction and the folding is that now in the folded protein, you can bring amino acids together that can carry out certain chemistry and that provide what's called the active site with which proteins now can fulfill these different functions. What we've learned over the last years by many, many different groups, is that this process of folding up a protein is very, very complicated. It's in most cases assisted by cellular machinery. There are folding intermediates that the protein undergoes before it reaches the finally folded state. Proteins are then degraded, so there's control how much of a protein is around, and worn-out proteins get tagged to be degraded. But these processes also can go wrong, and you can end up with unfolding of folded proteins. They can state into unproductive misfolded states from which they can be either rescued or they can be shunted into degradative pathways. And finally, proteins can aggregate into either amorphous aggregates, just think of a hard-boiled egg, or structured proteins that then can be toxic aggregates and are associated with many of the neurodegenerative diseases. And from there, they can be shunted into degradative pathways. This is a very, very active field, and it has already received 3 breakthrough prizes by work from Harry, Yoshinori, and Alex. And with Kazu Mori and myself, it's now 5, and I'm sure there will be many other aspects of this scheme here that will be covered in the future. These processes don't happen spontaneously. There are about 1,000 proteins in the cell dedicated to drive these different reactions to assure protein quality control. And many of the cell's proteins diminish. These control processes diminish as we age. And I think this is one of the big aspects over the next 10 years to see, can we maintain the health of a cell even as an organism ages by manipulating these processes. So our own entry into this field deals with protein folding in a particular intracellular organelle, a compartment, which is called the endoplasmic reticulum, where proteins enter that eventually make it to the cell surface. So all proteins that get folded there and then end up on the cell surface are secreted. Many of them are involved in these communication events between cells, and therefore tell one cell how it should behave in the context of an organism. As a consequence, this folding compartment of the endoplasmic reticulum is exquisitely well controlled, both in volume and in function. And we have figured out, both Kazu's lab and my lab, what these mechanisms are. There are sensor proteins sitting in the membrane of this compartment that recognize whether the system is in balance. Do we have enough protein folding capacity to handle the load of proteins that enter this compartment? And if unfolded proteins accumulate there, then these sensors become activated, and they drive a set of gene regulators that then downstream make sure that the balance is reachieved. The mechanisms by which this information is transferred here is incredibly beautiful. There are many, many new discoveries that we've made in these three different branches that all sense unfolded proteins. I won't go into the details of the mechanism, but they're really surprising and beautiful. This is my daughter, by the way. So we've uncovered some of these black boxes and know in incredible molecular detail how the information is being transferred. Downstream, then we have target genes that are expressed, and the idea of expressing these target genes is to bring the system back into balance, to balance the capacity and the load of this organelle. If this cannot be achieved, however, then cells make an active decision to enter a suicide program, to kill themselves, which, okay, is intellectually very interesting to ask for us. How can a cell decide, right? Can I fix the problem or should I kill myself? We have some idea now how this is going on with collaboration with colleagues at Genentech, that there is a balance between these pathways. Different timers are built in that allow the cell to first try to fix it, and then when time runs out, to commit suicide. And that is, of course, I mean, it's sort of logical that the cell sacrifices itself, because if you have misfolded proteins that end up on the cell surface, you can potentially create rogue cells that could become danger for the entire organism. But it's this life-death balance that the unfolded protein response provides that puts it in the center of many different human diseases. And this balance can err on either side, either on the cell protective side or on the destructive side. So in health, we think of the unfolded protein response as a finely tuned governor of protein health, so of protein homeostasis. In cells, it can either be a rogue cell executor that kills cells that we would rather have stay alive, and maybe the most intuitive example are inherited protein folding diseases, where the patients have a mutation in proteins that enter the endoplasmic reticulum, such as in retinitis pigmentosa, where the light receptors in our retina are encoded with mutations. So not only does the protein not work properly, but because the cell cannot fix the problem, it's an encoded mutation, it accumulates all the unfolded proteins, gets frustrated, and kills itself. So the entire retina degenerates by this suicide program, leading to this inherited form of blindness. We all eat a lot, so we're asking the beta cells of our pancreas to make ever-increasing amounts of protein, eventually exceeding the protein folding capacity, and the cells kill themselves. And of course, if one cell dies, the cell next door is asked to make even more protein, more insulin, so it becomes a downward spiral. But on the other hand, we also have the unfolded protein response acting as a rogue cell protector. And that happens in situations like viral infections, where viruses infect the cells. They amplify the endoplasmic reticulum to make more of their membrane proteins. And so if we could make an agent that could slightly shift this life-death balance, we could potentially have a very broad-spectrum antiviral agent, from which the virus could not mutate away because it's a cellular function that's being affected here. And finally, we have cancer. All cancer cells evolve They mutate constantly. They have very unstable gene expression programs. And not all the mutations, evolution is not directed, so not all the mutations help the cancer cell itself. So what happens then is that we accumulate a huge number of passenger mutations in cancer that are unfolded or misfolded proteins, imbalanced protein, imbalance in the proteome in the cell. And this is buffered by various proteins induced by these stress pathways, such as the unfolded protein response and other stress pathways. So cancer cells use these functions as an inappropriate, as a rogue cell protector. The challenge then, of course, is to identify in these disease settings whether we can affect this life-death balance, whether we can find a therapeutic window in which we can actually do more good than harm in a disease setting. And I want to just very briefly show you one of our approaches in which we screen for small molecules that affect these pathways. We now, in our lab and other labs, we have small molecules that affect each of these three branches. We can both up and down dial them. And this is one of these molecules which we isolated here. It's a beautiful molecule. There's beauty and symmetry. And we know precisely how it works. We have now a structure. It acts as a molecular clamp where it sits in the middle of two huge protein complexes and clamps them together to activate this protein into this nice structure. The surprise comes that this molecule, we can inject it into mice, is entirely non-toxic. There's no overt toxicity of it, which is really surprising because we're affecting one of these very central pathways that's present in every one of our cells. But by the molecular detail with which the molecule, this ISRIP molecule works, we call it ISRIP, is that it affects the process in a very subtle and beautiful way. A bigger surprise came out of a chance phone call I had with my colleague Nahum Sonnenberg at McGill University prior to publishing this work, so it really speaks for the value of open communication. And out of that phone call and conversation came suggestions for experiments that then led to the conclusion that this molecule that affects this pathway of the unfolded protein response acts in animals as a cognitive enhancer. So it allows mice to form better long-term memories. Entirely unexpected to me, putting us in a completely different field and showing the beauty of serendipitous discovery that comes out of curiosity-driven research. There are some profound implications from this simple result, and that is that these are normal mice, right? Yet we can enhance their cognitive function. So there must be a brake normally on. Evolution hasn't optimized this process, and there's an extra reservoir and cognitive capacity that can be tapped into. So this molecule ISRIP can release this brake. So then the question is, can we exploit this increase in cognitive capacity in disease? We collaborated with Susanna Rossi's lab at UCSF, who has a model. Susanna has a model of traumatic brain injury, where we basically test mice here in this assay, in this water maze. So the mice have to swim to this hidden platform in one of the arms. They orient themselves with visual clues around the room, the same way we will have to orient ourselves on the Stanford campus here. And we basically train them in this task, and we measure the time, and we measure the number of errors the mouse makes swimming into the wrong arm. And this is the result here. 2 weeks later, when we look how much do they remember about what they've learned, control mice make, on average, 1 mistake, whereas mice with that have received this brain injury make 3 to 4 mistakes, and that's basically a random exploring. So they don't learn. They are demented, permanently demented in this situation. If we inject the drug, the amazing result is that these cognitive defects are entirely restored. So this gives us hope. So ISRIP corrects the cognitive defects after brain injury, even if we give the drug 4 weeks after the injury has actually been inflicted and these permanent cognitive changes have occurred. So these results open new paths to how memories are stored and lost. And they allow us to think about potential therapies in diseases which have cognitive defects. In addition to traumatic brain injury, we have many of the neurodegenerative diseases that are connected with cognitive dysfunction. And then as it becomes increasingly apparent to me, it's also a process that occurs with aging. So hopefully the development will keep pace with us getting old. This is, of course, fantastic news for all the mice in the audience. We have no idea whether any of this will translate into human physiology. Mouse brain and human brains are quite different. But as I said, we are quite hopeful. So that brings me to the end here. What we try to do then strategically is to understand the precise molecular mechanisms that control the fate of cell proteins. We would like to modulate these mechanisms in various disease models and translate then our knowledge from animals to humans, and then assess the safety and utility for therapeutic interventions in various clinical trials. And this is sort of a continuum between academia and biotech, where the collaborations are incredibly powerful, especially in the middle range of this, and we have very good connectivities in the Bay Area here to push this to the next step. 10-year dream is that we will use this knowledge, we as a field, to develop new cures, novel cures, for many of the diseases that I mentioned to you, for cancers, cognitive disorders, and other diseases, by targeting these Achilles heels of cell quality control going awry, and develop preventive measures that keep our proteome in cells healthy, and therefore nudge our society a little bit more from the current strategy of basically dealing with diseases as they occur, to become more proactive and preventive, so going from sick care to health care. Schiller used this, Friedrich Schiller, a German philosopher, used this beautiful word, edle Wissensbegierde, so the noble quest for pure knowledge, which sort of describes beautifully what we do in terms of curiosity-driven research. We started our work as entirely disease agnostic, and the broad spectrum, I think, justifies it. It touches many different ways. It was not predictably translational, yet our funding agencies sort of ask us to justify what some of our knowledge will be useful for in the long run. And finally, it is entirely open-ended what we do. There is no end in sight. And this is, we have a wonderful future ahead here of discovering more. And because it's so open-ended, there is room for all the young people entering the field. As Einstein said so beautifully, as the circle of light increases, so does the circumference of darkness around it. And of course, it's in that range of darkness. The more answers we get, the more questions we generate. And it's in this circle of darkness then that all the next generation will find their careers and then make new discoveries. With that, this is the work I'm being honored here for, is, of course, I have to credit the many, many people over the years who've worked and dedicated their work in the lab, and the large number of collaborators who so generously shared their expertise with us. Thank you so much.
Excellent. Some time for questions. May I take you right there?
Sorry, I have a sort of two parts. I'm really curious about what your current working hypothesis is for how the ISRIB drug affects cognition. And then I'm also curious about what is it that Nahum Sonnenberg said to you that gave you the clue to go and investigate this angle?
Yeah, so let me start with the second question. So Nahum had done, Mauro Costamaccioli was a postdoc in Nahum's lab and did genetic work that the very process that we've been affecting with the drug here is also affected and gives cognitive advantages in heterozygous mice that partially block this pathway. The pathway that's being affected here is a translational control pathway by which the phosphorylation of a translation initiation factor, which under certain stress conditions inhibits protein synthesis, is now alleviated by overcoming the negative effects of the phosphate being added to the ERF2. We think that then translates into the proteome, the composition of proteins in these neurons that are affected now in the learning process in a way that synaptic plasticity is being affected. And that basically you are able again to form stronger synapses. And that of course goes all the way back to Eric Kandel, who showed that learning in aplesia requires active translation to go on.
Hey Peter, I have a question for you, and I was just wondering, what is the relationship between the death pathway that's caused by unfolded proteins generated from the ER with the mitochondrial, pathways that cause cell death?
Yeah. So the activation of the unfolded protein response leads to activation of the apoptotic program in the cells. If we unfold proteins in different compartments, such as the cytosol, we also affect the unfolded protein response, because now we are backing up degradation systems, and therefore proteins that normally get folded in the ER but misfold, they are shipped back into the cytosol for degradation. And in that case, now if you don't have enough degradative capacity in the endoplasmic in the cytoplasm, then this process of moving proteins back into removing them from the cell is blocked, leading to an increased susceptibility to cell death. So all these pathways are connected. So it's a huge network that spans compartmental differences.
Question here? Oh, right here in the back.
So Peter, you found this drug which can inhibit this protective pathway, and you then administered it to mice, and you reported the amazing part that it's not toxic.
Yeah.
It's actually quite good. So isn't there a paradox there? You've inhibited the normal pathway that you thought was one that was going to be protective, and inhibiting it actually proves to be the protection?
Yeah.
Did I get that right?
I love paradoxes. The reason is that we are not inhibiting entirely. So first of all, we are not inhibiting protein synthesis, but what we are doing is we are inhibiting an inhibition, right? So the pathway has to be on in order for the drug to affect anything. So if the stress pathway is not on, it doesn't do anything. The second part of this is that if you cannot do the signaling through the pathway, which we can test genetically, for example, by just blocking this phosphorylation event I mentioned, then the mice are embryonically lethal. So these pathways are required to function. But what we are doing here is we are affecting a very subtle difference in the middle, where the dose-response curves are affected in such a way that we actually have incremental. So basically, in the heterozygous mouse that shows this better learning behavior in the genetic models, we only make half as much of the inhibitor. And what we find is that if we have a lot of phosphorylation going on, this pathway is still protective. So I think we can do it both ways. We can still keep the protection at high induction of the pathways, but what we're doing is we're modulating it in a middle range of a concentration of the inhibitor. There's a lot to be learned, and I think it's in the dose of the response that is being affected here.
Okay, very good. Let's thank Peter again.
Thank you.
Thank you. Thanks so much.